Hello, everybody. Good afternoon and welcome to our grand rounds. Actually, today is a special day. It's our Research and Scholarship Day in which we honor some of the research and scholarship that are done in our department. Unfortunately, because of the pandemic, we don't quite have our poster session and guest speaker like we've done in days past. But we thought we'd try a truncated version of that today. And so let's see how this works. So first off, we usually on this day like to congratulate the Award of Excellence in Pediatrics to the graduating medical student who has done quite well in pediatrics. This award comes from the South Dakota School of Medicine, Department of Pediatrics, as well as the South Dakota chapter of the American Academy of Pediatrics. And our award winner is Dr. Rebecca Mabey. So congratulations, Rebecca. And what's exciting about that is Dr. Mabey also is joining the University of South Dakota Pediatrics residency program in a few weeks. And so it's good to have Rebecca join our club. So with that in mind, we had 7 people submit pediatric research abstracts this year. And I'd like to just mention everybody's name. We thank Dane Hegdahl, Dave Arendt, and Benson Shu for infant with acute bilateral pneumothorax during air transport. Caitlin Dorn for Diamond Blackfan anemia, a case report and review of the literature. Nathan Bleisseg and Hannah Williams and Shelby Jefferson and Jessica Messersmith for the impact of COVID-19 pandemic on newborn hearing screening programs in the Western states. And again, we thank Benson Shu along with Andrew Reuter, Jen Shu, and Katie Patrick for the impact of political affiliation on COVID-19 beliefs and vaccine acceptance in the US. We hope to have them present their abstracts later on this year, but today we focus on 3 presentations, and I will let them introduce themselves as they begin. We will start with compliance rate of pediatric lipid screening at a rural independently owned primary care and multi-specialty clinic. And this was submitted by Sarah Lehman, Lauren Van Hove, and Michelle Olson. So please take over.
Hello, good morning, everybody. Can you hear me?
Yes.
All right, sounds good. So like he said, my name is Michelle Olson. I have Lauren Van Hove and Sarah Lehman with me. We are all 3rd-year medical students here at USD. Today, we are going to be presenting our quality improvement project, which was titled Compliance Rates of Pediatric Lipid Screenings at a Rural Independently Owned Primary Care and Multi-Specialty Clinic. So the background for our project started in 1998, which is when the American Academy of Pediatrics first recommended pediatric lipid screenings. At that time, the recommendation was to only perform a total cholesterol screen for high-risk children ages 2 to 18. As time progressed and childhood obesity increased in prevalence, this recommendation was expanded to include more criteria under the high-risk definition. The screen was also changed to be a fasting lipid panel for those children 2 to 10 years old with recurrence based upon the findings. Finally, in 2011, the National Heart, Lung, and Blood Institute made the recommendation for all children to be screened with a fasting lipid panel between the ages of 9 to 11, and again between 17 to 21. The first step of our project was a literature review to determine what is the national compliance rate for the screening recommendation. Unfortunately, as we'll explain here shortly, we found there are not many studies done covering the compliance rate of the lipid screenings. One publication we did find was a survey done by the American Academy of Pediatrics in 2013 to assess practicing pediatricians' views on the 2011 guidelines. While nearly 2/3 of pediatricians were aware of the guidelines, less than 1/3 actually were practicing universal screenings. Instead, the majority of pediatricians were conducting a selective screening based upon patients they considered high risk.
For our methods, what we did was a cross-sectional study design to assess the compliance with the universal lipid screenings in patients 9 to 11 and 17 to 21 in accordance with the 2011 NHLBI recommendations. So we retrieved our information from the electronic medical record at the clinic that we were looking at, and the inclusion criteria were that for the 9 to 11 group, that patient had to complete age 9 to 11 during our specific time frame, and for the 17 to 21, they had to complete that entire time frame during our specified time frame. They also had to have at least one well child exam or annual physical from January 1st of 2014 to December 31st, 2019. For our results in table 1, if you look at that, you will see that it details the percentage of patients who either received a lipid panel screen or a total cholesterol screening in either of the groups. The percentage of patients that received a lipid panel screen were very low in both age groups at 3.3% for the 9 to 11 and 4.2% for the 17 to 21. However, the percentage of patients that received a total cholesterol screen was higher for the 9 to 11 age group at 13.7% compared to the 3.4% for the 17 to 21 age group. We added those together, so the total percentage of patients that received either screen for the 9 to 11 was 17% compared to 7.6% for the 17 to 21. Of the patients that did receive the lipid screening, 31.8% in the 9 to 11 age group and 80% in the 17 to 21 had at least one abnormal lipid level. In table 2, we wanted to ask, was a lipid screening offered by the physician? Were people just turning it down if it was offered? And so we went through all of the notes in the charts to see if a lipid screening was in fact offered and declined. If there was no mentioning of it, then we marked that as no lipid screening offered. And so the first column shows the percentage of patients with no lipid panel or cholesterol level performed, which was 83% in the 9 to 11 and 92% in the 17 to 21. And of those, 98.9% in the 9 to 11 were not offered the screening based on the notes, and 99.1% in the 17 to 21. Of those offered but declined, it was 1.1% in the 9 to 11 and 0.9% in the 17 to 21. We tried to document reasoning for a decline. Obviously, we had very few declines, and there was one parent requested just to have it postponed, and some of them didn't have reasons documented.
As previously noted by Lauren, our compliance rate with universal pediatric lipid screening was found to be very low. So we wanted to assess compliance with a more risk stratified model. Based on the information that we were able to retrieve from the EMR, we sorted our patients by BMI, with obese category classified as greater than the 95th percentile, overweight as greater than the 85th percentile, and normal weight as less than the 85th percentile. Although we did see greater compliance with lipid screening in these high-risk individuals, there was still significant room for improvement. So if we look at table 3 here, we can see that among the patients who would be the highest risk based solely on BMI, we see that only 11.1% of the 9 to 11-year-olds were screened with either screening, and 13.3% of those in the 17 to 21 age group. So we definitely have room for improvement here based solely on a BMI risk stratification scale. Overall, our study revealed low compliance with the 2011 NHLBI universal screening and the BMI-based risk stratification lipid screening. There are multiple reasons that this could have occurred. One of those would be lack of awareness of the changing guidelines. So as Michelle detailed in the introduction, there have been numerous changes with very large time gaps in between to these guidelines, and that can be difficult to keep track of, as well as a variable acceptance of the NHLBI guidelines by professional medical societies. So for example, the USPSTF found insufficient evidence to endorse the universal pediatric lipid screening, but the AAP fully endorsed the recommendation. And that can be hard for physicians to decide which screening guidelines to follow. As previously discussed, there is some lack of research regarding efficacy of screening and health outcomes of early intervention in pediatric dyslipidemia, especially given the fact that there is so much variability in the suggested therapeutics for pediatric dyslipidemia. Most pertinent to our study, and because this was an HQIP project, the lipid screening guidelines are not included in the anticipatory guidance of the EMR at the clinic that we analyzed. And so physicians aren't receiving any reminder that between these age groups, they should be screening for cholesterol levels. One thing I just wanted to add to our presentation is that there are some limitations to our study. As you can see, there were very few patients who actually received a lipid panel or total cholesterol screening, which then skewed our results for how many patients were affected by dyslipidemia in the population that we studied. And so we just wanted to point that out. Future studies regarding this topic include assessing the compliance with universal screening at a national level, as well as evaluating the cost effectiveness of screening and the impact on pediatric health outcomes. Next, you'll see our resources, and then are there any questions?
And start.
I'll ask a question, if that's okay. Can you guys tell me what lipids they're actually screening for on these panels? I'm not a clinician, so just kind of curious here.
So the full lipid panel includes, and correct me if I mess this up because I do this sometimes, but it should be a total cholesterol, LDL, which is the bad cholesterol, HDL, which is the good cholesterol, and then we also look for triglycerides, which is a part of fat in the blood. So that's what it's looking for. Just kind of a fun fact that we looked up for our presentation is that the average lipid panel actually costs $130 or so. And so that could be partially reasoning why clinicians aren't ordering it in low-risk patients as well.
Anyone? Any other questions? Okay. Well, thank you very much. And I look forward to seeing your presentation for next year. Okay. Anyway, take care. Next we have, well, Janet, who do we have next? Why, we have Abigail Varsho from the Bach Lab, who is going to talk to us about pre-ovulatory CoQ10 supplementation to prevent mitochondrial dysfunction in offspring born to mothers on high-fat diets. Okay, take it away, Abigail. Who are you and what are you up to?
So my name is Abigail Varsho. I'm a first-year PhD student in the Bach Lab at Sanford Research through USD. And my project, as mentioned, is peri-ovulatory CoQ10 supplementation in order to prevent mitochondrial dysfunction in offspring born to mothers on high-fat diets. There we go. So obesity is known to be associated with detrimental fertility outcomes, including menstrual dysfunction and ovulation, infertility, miscarriage, pregnancy complications, as well as programmed cardiometabolic disease for the baby. Interestingly, over 38% of women around reproductive age are considered obese, but even without obesity, a high-fat diet can incite these reproductive consequences as well. A definition of a high-fat diet is actually anything greater than 30% of a caloric intake as fat. And interestingly, the average westernized diet is around 35%. And these consequences extend beyond the mother to her developing baby as well. So when we think about the maternal diet and its effects on the fetus, we think about something, the DOHAD, or the Developmental Origins of Health and Disease, which was popularized by a British epidemiologist, Dr. David Barker. And he hypothesized that the environmental exposures from the mother, including undernutrition and overnutrition, during early development can cause lifelong changes leading to a greater risk of adult disease. And when we specifically look at maternal and fetal overnutrition, like diabetes, obesity, and a high-fat diet, this can increase the lifetime risk of cardiometabolic disease for the offspring in the form of overgrowth, cardiorespiratory disease, and metabolic syndrome and heart disease in later adulthood. Importantly, some of these effects can be passed on to subsequent generations through inheritance of maternal mitochondria and through epigenetic changes that can pass from one generation to the next, which really perpetuates this cycle of cardiometabolic disease. The Bach Lab's goal is to really understand the cause of this developmentally programmed disease and to develop translatable interventions in order to improve the lifelong health of these high-risk babies and for their future generations, and to lower this overall burden of cardiovascular disease in the future. So to do this, we developed a rat model of fetal overnutrition, and we're looking over the course of their lifetime, starting from postnatal day 1 all the way up to late adulthood, or 12 months. And our results showed that a high-fat diet actually increases the risk of stillbirths, neonatal deaths, newborn and cardiac and adult cardiac disease. And this is happening through mitochondrial dysfunction, oxidative stress, and an altered cell fate. So this mitochondrial dysfunction and oxidative stress is actually working through the mechanism of the electron transport chain within the mitochondria. And when electrons aren't able to flow easily through the electron transport chain, we get less APD produced. And this really causes an inability to meet the cell's energetic demands. And in addition, this dysfunctional mitochondria can increase the levels of reactive oxygen species. And when these exist in excess of the body's antioxidant capabilities, this will really cause oxidative damage to lipids, proteins, DNA, and RNA. And it can cause mutagenesis and transcriptional changes, well, not only in the mitochondrial DNA, but in the nuclear genome as well. And then in order to prevent these effects, we want to know whether the mitochondrial dysfunction is inherited through the maternal mitochondria or if it's due to in utero exposure. So we believe that mitochondrial dysfunction is really contributing to ovarian aging and a diminished oocyte reserve. So as you can see in our picture, poor mitochondrial function results in poor energy production because of less ATP being produced, and an increase of reactive oxygen species production, which can cause cellular damage. And this damage can be inheritable through perturbations in the nuclear and the mitochondrial genome. And this can be passed on to the developing blastocyst, which will affect the oocyte development as well if this fetus is female. So because of this, we hypothesize that a maternal high-fat diet causes premature aging, embryonic mutagenesis, inheritable metabolic dysfunction, and these are all happening through mitochondrial mediated mechanisms. So we predict that a dietary supplementation with a mitochondrial antioxidant, CoQ10 or coenzyme Q10, will help to mitigate these effects. Importantly, the oocyte and the early development of the embryo is highly dependent on the quality and the quantity of the mitochondria. So you can see that the primordial germ cell is developing in utero and then it remains quiescent until puberty. And then during folliculogenesis and up until ovulation, the mitochondrial DNA amplifies about 1,000-fold in order to support the energy of fertilization, division, and implantation into the uterus. And this is very important to note that this mitochondrial DNA amplification is actually at risk of amplifying any mutations associated with dysfunctional mitochondria right up to ovulation. So this can create a greater risk to the developing offspring. So when we have this combination of energy failure and oxidative stress, this is really playing a detrimental role to the female reproductive system. And this can be caused by a variety of conditions and syndromes. Advanced age is one of the most common and well-studied and well-known. Although when we look at fetal overnutrition, like obesity, high-fat diet, and diabetes, these can incite some of the same phenotypic responses. And other conditions such as endometriosis, polycystic ovarian syndrome, and various mitochondrial diseases can show this as well. And these lead to detrimental effects in the offspring, including aneuploidy, fertilization and implantation failure, pregnancy loss, oocyte reserve depletion, and metabolic disease risk in the offspring. So why coenzyme Q10 or coenzyme Q? So if we remember back to our electron transport chain diagram, we can see coenzyme Q here, and it's really a powerful antioxidant to help sequester any reactive oxygen species produced, as well as helping transport electrons through this electron transport chain in order to assist in ATP production. In addition, we have previously found that in the offspring of our diabetic mothers that were on a high-fat diet, they had a downregulation of an ADCK3 gene, which actually encodes a protein involved in the synthesis of this coenzyme Q10. So the main goals of my project are to quantify the CoQ10 levels within the serum and the ovaries of a control and a high-fat diet of our rats with and without this periovulatory supplementation. And then we want to analyze the effects that this supplement has on not only fertility outcomes, but also embryo quality and viability. So for the methods of this project, I will put female rats on a control or a high-fat diet for at least 28 days. I'll then add a CoQ10 supplement to the diet for at least 2 ovulatory cycles. I'll start breeding, and then at postcoital day 4, we'll isolate the embryos, and we expect to find them in this blastocyst stage here. So although our studies are ongoing and some of our numbers remain low, our preliminary data suggests that this high-fat diet is inciting oxidative stress within the ovaries of our mother rats. So we can see this through lipid damage and DNA damage. So first, looking at lipid damage, we ran a MDA or malonaldehyde assay, and this analyzes the lipid peroxidation. And we can see that overall, there's an increase in the high-fat diet rats compared to the control. And then when we look at our DNA damage, we can see first through an 8-oxo-G assay, this occurs when a hydroxyl radical interacts with a guanine, causing DNA mispairing. And this can actually cause mutagenesis as well as a decreased rate of transcription. And we can see there's a trend towards increase in our high-fat diet rats as well. And then to see if this oxidative damage is causing mutations, we looked at specifically the mitochondrial DNA within the ovaries. And this is a type of mitochondrial DNA deletion screening assay. And we can see that we can look at the mitochondrial deletions or fragmentations that indicate mitochondrial DNA damage, which over time will cause metabolic dysfunction. And you can see that the DNA band count here correlates to DNA mutations. So we can see overall there was a significant increase in our high-fat diet. And interesting to note that in primer 4, there is a trend towards an increase. And this actually encodes for a protein ND1 of the respiratory complex. So as we're assessing embryo quality, since we isolate our embryos at postcoital day 4, you can see here they call it embryonic day 4, same thing. And you can see that we are expecting to see these nice mid-stage blastocysts. However, when we have poor quality within our embryos, this will result in a reduced or an arrest of maturation to more advanced stages, as well as apoptosis of some cells within the embryo. So as we have been validating some of our methods and finding the best yield, all these blastocysts I harvested on the postcoital day 4. And you can see here our 10 to 12 week rats. In all 4 of these images, we have very great looking blastocyst stage embryos. And you can see that there's this faint outer layer is the zona pellucida, and it's nice and round. Right inside of this, you have a tightly packed cells, that's the trophectoderm. And then we have this mass here, which is the inner cell mass. And then as we move on to increase age, we see at 43 weeks and 69 weeks that the blastocysts have not matured into the embryos have not matured into the blastocyst stage. And this makes sense since rats do enter menopause around 65 to 78 weeks. So you can see this is the decline of their fertility. And as we look, even though this is only increasing age, we do expect to find some of the same things and same phenotypes happening within our high-fat diet rats. So just from some of our preliminary results, this is a 12-week rat that was on a high-fat diet. And you can see in comparison to the 12-week rat here, which this should be the peak of their fertility, you can see that it has not developed quite to the blastocyst stage. It appears to be in an 8-cell stage or maybe a morula. And if you look at the zona pellucidas, they're more of oval shape or not that nice round shape as we see in the control 12-week rats. In addition, we went on to culture these embryos to see if they would develop over time further, and we validated some of this using the some embryo viability assays. So we can look at trypan blue, which will stain any dead cells blue, so we can see that this is viable. And then our live dead fluorescent assay, the green represents alive cells, and then the red represents dead cells. This is just another way to confirm. And then to assure that our CoQ10 diets are actually increasing the levels of the serum and within the serum and ovaries of our mother rats, we assessed levels of 2 different dosages for 10 days or 2 ovulatory cycles. So we can see there's a trend towards a dose-dependent curve, and we chose 10 parts per million and 95 parts per million for our supplementation diet. And we chose our highest being 95 parts per million because of its clinical relevance to about 600 milligrams per day as a supplement for, which has been seen in some clinical trials that has looked at women with advanced age that have had failed IVF treatment. And then we also have our results for our ovary as well. We do, to determine if we can gain further increases at the tissue level, we are going to be extending the time on the supplement in order to be more comparable to that of a human ovulatory cycle, since rats only have about a 5-day cycle compared to the 28-day cycle of a rat. And we also plan to measure CoQ9, which is an equivalent form that rats primarily use. So in conclusion, we have determined that high-fat diet is inciting oxidative stress, which impacts the maternal ovary and its DNA. It affects pregnancy outcomes, and it can program cardiometabolic disease risk to the developing offspring. And we believe that our CoQ10 supplementation will serve as a potent mitochondrial antioxidant with therapeutic potential to really mitigate these effects through advanced family planning to target a very specific population, as well as enhancing fertility by reducing this oxidative stress, mitochondrial dysfunction, and mitigating this cardiac risk in our offspring. So with that, I'd just like to thank Dr. Michelle Bach and the entire Bach lab, including Tricia Larson and Ty Gandy, as well as the Sanford Animal Resource Center and the core staff and our funding sources. And with that, I will take any questions.
Well, thank you, Abigail. Anyone else with questions at this moment? That was very good. Abigail, I do have a question. So have you started showing, have you started working on intervention then? I mean, you've got the control, you've got the, you've shown the pictures. What do you have with the intervention?
Yeah, we're really on our preliminary data at the moment. We're in the process of growing up some of our rats. So unfortunately, we aren't able to show our supplement right now, but we have some rats that are on high-fat diet currently that we're hoping to treat soon.
Okay.
Hey, Abigail, I'll ask a question too. So is CoQ10, is that included or part of kind of prenatal vitamins that a lot of people would normally take, or would this be an additional supplement that they would have to, someone would have to go out of their way to take?
Yeah, that's a great question. As far as I know, this is not included within the normal like prenatal vitamins that someone would recommend. I know they, it actually has been, it's actually sold over the counter currently, and they market it for different cardiometabolic disease as well. So I think right now it'd be a separate thing that someone would take.
I guess another question would be, so you're studying ovaries and the production of the blastocysts or whatever. Any research into the male counterpart?
Yeah, we do get that question. Right now we haven't looked very much at the male or the sperm involved in it, but we have mainly been looking at the female, since very low amounts of male mitochondria are inherited, and that's kind of our basis. But we do plan on maybe doing some assays with the female or with the male testes, just to look at their oxidative stress as well and see if that's contributing at all to it.
Okay. Great. I'm looking at chat to see if any questions there. No. So Abigail, thank you very much. And the pressure's off. And now we put the spotlight on Ellie Anderson, who will talk about sterols facilitate curvature generation, which I would like to hear defined, necessary for efficient endocytic trafficking within disorders of cholesterol synthesis. Take it away, Ellie. And please
thank you very
introduce
much.
yourself and what are you up to?
Absolutely. Can you hear me okay? Excellent. Okay. Well, thank you for having me. My name is Ellie, and I'm a 6th-year MD PhD student, or a Pillar 2 medical student here at USD. And today I'll be sharing with you some of the work from my graduate training, which was conducted under the mentorship of Dr. Kevin Francis at Sanford Research. And so our lab studies rare pediatric conditions which result from genetic mutations within the cholesterol synthesis pathway. And the focus of my research has been on how these altered membrane lipid composition influences cellular trafficking. And so I've titled my talk, Bending Membranes, as we've visualized an important role for cholesterol in facilitating curvature within clathrin-mediated endocytosis. And I'll unpack what that means over the next couple slides. So I have no disclosures to make. And I'll begin with just a brief introduction into disorders of cholesterol synthesis. So the hallmark of these conditions is both a decrease in cellular cholesterol, as well as accumulation of these various precursors in their metabolites. And so as such, a major challenge in this field is distinguishing between these shared effects. And for today's talk, I'm going to focus predominantly on the terminal end of this pathway and discuss Smith-Lemli-Opitz syndrome. So Smith-Lemli-Opitz, or SLOS, is the most common of these rare diseases, and it's caused by mutations within the gene DHCR7 with an autosomal recessive inheritance. And so clinical severity will generally correlate with residual enzyme activity, such that these clinical presentations can exist on a really wide spectrum, from mild cognitive impairment all the way to prenatal or perinatal death. And as you may know, SLOS manifests as a broad malformation syndrome, and this can include features like cleft palate, syndactyly, cardiac defects, holopresencephaly, which are thought to involve the sonic hedgehog signaling pathway. And the CNS is particularly vulnerable to cholesterol depletion due to the presence of the blood-brain barrier and thus dependence on de novo synthesis, such that most of these patients exhibit autistic behavior, developmental delay, intellectual disability, and sometimes seizures. And unfortunately, cholesterol supplementation is the only available therapy for these patients, and randomized control trials regarding its efficacy have been largely disappointing. And this leaves baseline cholesterol levels as the best indicator for a patient's developmental trajectory over time. And similarly, preclinical gene therapy within SLOS has been relatively disappointing. And so thus there's a strong need to better understand the mechanisms of disease in order to develop new therapeutic strategies for SLOS, which is the motivation for our work. And so we became interested in endocytosis, which is the process by which cells are internalizing nutrients, cargo, and signals, by looking at the subcellular distribution of cholesterol and its functional role there. So one statistic that I like to share is that the plasma membrane is estimated to account for up to 60 to 90% of free cholesterol. And it's thought that these strong lipid gradients, which are maintained between or across these organelles, may provide a self-organizing directionality to some of these pathways. And in addition to this, cholesterol's role, its structural role in the membrane is extremely relevant for these highly dynamic remodeling processes like endocytosis. And so because of this, we focused our efforts on clathrin-mediated endocytosis, which I'll call CME for short, which is the principal pathway utilized for receptor-mediated signaling. And so this is a depiction of CME progression. And basically, there's a structurally unfavorable transformation that has to take place within the membrane for these cargo-laden vesicles to get internalized. And during this process, all you really need to know is that clathrin assembles at the plasma membrane. That gives it kind of that distinctive honeycomb-like appearance. And then at late stages of vesicle formation, we get dynamin recruitment to this neck, which allows for the scission event itself. And so our hypothesis was that CME may be disrupted, possibly at a stage of membrane bending. And so one method that we employ to model SLOS is through small molecule inhibition of the synthetic pathway. So on one hand, you have your well-known statins, which will deplete sterols chronically. And we also have methyl beta cyclodextrin, which is this ring structure, and it can strip sterols away from the membrane in an acute fashion. And then we also have a couple non-competitive inhibitors, U18 and AY, which affect either the reduction of the side chain or the ring structure respectively. And so importantly from this slide, AY is our model for SLOS. So next, we wanted to monitor endogenous clathrin trafficking. And to do this, we used CRISPR-Cas9 to knock in a fluorescent tag to the clathrin light chain, so right here. And here's a movie of what we found. So this is taken right across the midplane of the cells. And within this first panel is a control condition where the cells have to synthesize their own cholesterol. And hopefully you can appreciate the rapid trafficking throughout the cell body. However, with our AY model, we see stagnation of clathrin at the plasma membrane, which allows you to kind of distinctly see these cell-to-cell junctions. And then moreover, this is an effect that we can readily rescue by directly delivering cholesterol back to the plasma membrane using cyclodextrin as a carrier. We can also quantify CME activity by monitoring the uptake of fluorescent cargo, like transferrin, which gets exclusively internalized through CME. And so again, if we can compare our control condition to our SLOS model, instead of these scattered internalized puncta like we see in controls, instead, we find transferrin bound around the cell membrane. And this is an important point because it allows us to confirm that the receptor can traffic to the membrane and bind transferrin, but that it's failing to undergo internalization. And then over on the right, through correlative studies, we can demonstrate that CME activity is most closely related to total sterol abundance. We can also use this same technique to look at CME dynamics in patient fibroblasts. And this was taken across from subjects across the phenotypic spectrum of SLOS. And down here, if you compare it to our unaffected control, which is in black, CME was greatly reduced in all of our SLOS samples upon culture in these cholesterol deplete conditions. And notably, even within serum-rich conditions, fibroblasts from the more biochemically severe lines trended towards diminished CME activity as well. And this suggests that exogenous lipid supplementation may be unable to fully rescue CME function, perhaps because LDL internalization is also a CME-dependent process. And so next, we wanted to look at the mechanism involved in this dysregulation. And to do this, we used a technique called polarized turf microscopy. And basically, this allows us to monitor membrane curvature generation in relation to recruitment of CME proteins. So just very briefly, turf allows us to visualize events taking place on the plasma membrane. And then the polarization piece of this allows us to selectively excite membrane that's been labeled with a dye in either the vertical or the horizontal orientation. And then we can take the ratio of these images to give us a readout of curvature in real time. And so for these studies, we used a cell line in which clathrin and dynamin have been endogenously tagged, as you see in these first two panels. And then our curvature signal is in this third panel. And here's what this looks like. And then through tracking analysis, we can associate each of these clathrin events with both curvature and dynamin recruitment over time, like you see in this kymograph above. And so in this case, we get membrane bending occurring as the clathrin-coated pit assembles. There's a flash of dynamin, and then they all disappear together. And so utilizing this technique, we were surprised to find a graded response in membrane bending, which corresponded to the degree of sterile depletion that we achieved. So just to orient you to these graphs here, this is space, and this is the time dimension, which is 6 minutes in length. And within our control conditions, you can see these many flashes of dynamin, and then within the merge channel, maybe a dozen or more CME events taking place. And this is in contrast with our SLOS model, where sterile levels have been depleted to approximately 80% of controls. And here we see these long-lived clathrin tracks, which have a high degree of curvature associated with them, but a delay in dynamin recruitment, as you can see down below in the merge channel. And then finally, with our severe depletion with cyclodextrin treatment, here cells are depleted to approximately 40% sterile levels compared to controls. And this nearly completely shuts down CME, as you can see by these long-lived clathrin tracks with little to no curvature and no dynamin recruitment. And over on the right, we can actually quantify this. And here we can see that with sterile depletion, we get increased clathrin lifetimes. And then by using dynamin as a marker for completed CME events, we find that the number of productive events per cell area is dramatically reduced during sterile depletion. And so next, to validate these live cell imaging studies, we performed platinum replica transmission electron microscopy on these cells under identical conditions to the studies shown before. And so here we scored these clathrin structures into one of 4 categories. And within controls, this is seen in either black or the gray. Here the cells are cultured with cholesterol-rich serum, and here the cells are synthesizing their own cholesterol. We see this relatively rapid progression from flat to spherical pits. And this is in contrast to mild sterile depletion, as in our SLOS model or our statin treatment, which show an increased abundance of these shallow and domed pits, predominantly at the expense of spherical ones. And then finally, with our cyclodextrin treatment, we see very flat or shallow pits, which is in really good agreement with our live cell imaging studies. And then over on the right, we can also quantify the area of the plasma membrane that's been covered with clathrin, which increases from 3 to 6-fold, which demonstrates that these clathrin-coated pits are accumulating on the plasma membrane with sterile depletion. And so if you take all of this together, we think our findings provide structural evidence that sterile abundance influences the progression of CME events by altering membrane bending dynamics at multiple stages of curvature generation. And so we think that this is consistent with a biophysical mechanism by which cholesterol reduces the energetic costs associated with the initiation of membrane bending, as well as formation of this vesicle neck, as illustrated in that model. And so why is this interesting, and why does it matter within the context of SLOS? Well, there are a number of ways in which endocytic deficits could be contributing to mechanisms of disease. And ongoing work in the Francis lab involves understanding neurodevelopment within the context of SLOS using patient-derived induced pluripotent stem cell models in both 2D and 3D culture. And some of the most striking features we observe is premature differentiation, which is shown on the top right here, and as well as premature differentiation and changes in cell fate specification, which is what I'm showing down below. And CME is really important for both of these processes in that CME regulates signaling cascades involved in the maintenance of pluripotency. And this could be a contributor to the microcephalic phenotypes that we see as I'm showing down below. Rapid endocytic trafficking is also extremely relevant to functional neuronal activity, including neurotransmission and synaptic plasticity, which are processes important for cognition and learning and memory. And so over on the right, I'm showing a comparison of synapses, which were taken from a SLOS and a gene-corrected iPSC-derived neuronal differentiations. And hopefully you can appreciate that these contact sites within the SLOS model are somewhat distorted and have qualitatively fewer synaptic vesicles compared to our controls. And then the final point I'd like to make is that CME dysregulation may also be relevant to cholesterol supplementation by limiting LDL bioavailability, as well as shedding light on some of the challenges associated with gene therapy. And so it's our hope that this work will help us better understand the pathogenesis of these diseases, which will help us recognize new ways to approach treatment within these disorders. And so with that, thank you for listening and thank you for the opportunity to share our work with you today. I would be happy to take any questions.
Ellie, I have a question. Good job. Of course, your work has been amazing in the Francis Lab, and their ongoing work is amazing too. I have a question about whether CME plays a more important functional role within different types of cells within the brain or certain areas of the brain, specifically in the context of autism, and what you know about that or what your lab's doing to figure that out.
Certainly, yeah. So one of the things that we're interested in looking at is how this affects different neural progenitors and precursors, so astrocytes versus neurons. And there's a lot of interplay, especially relating to cholesterol metabolism within that dynamic. So we think that neurons can produce some of their own cholesterol, but it may have to be a majority of it may be trafficked from astrocytes as well. And so that's one piece of this that we're really interested in as far as the vulnerability goes. So if neurons aren't synthesizing it themselves and are dependent on that trafficking mechanisms, they may be at an increased risk of depletion. Yeah, and I think there's a number of ways that this may fit in with autistic phenotypes, in that a lot of the genetic studies have implicated different cellular trafficking pathways. And so that may be one way in which, kind of this paradigm with cholesterol dysregulation may be tied into some of these, learning and behavioral processes, as well as neurodegenerative changes too. So,
Thank
thanks for the question.
That was very good, Ellie. Joe Zennel, I do risk showing I may not have fully understood what you presented by asking the question, but so you show how important the clathrin-mediated cyst process is.
Mm-hmm.
How do you intervene at this point? You suggest gene therapy, which implies that it's too late for people already born with this issue, or do you go after supplying clathrin? The cells. What's your guess, or what do you think is the approach to go?
Absolutely. I think the major challenge with treating SLOS is that there's both, structural changes that are occurring during development, which are going to be really difficult to treat because that would have to be treated in utero, as opposed to some of these functional aspects like neuronal activity and synaptic trafficking, which you may be able to intervene at a later stage. And so I think it really comes down to, I'm interested in cholesterol bioavailability, meaning that, cholesterol through the diet is only available through LDL uptake. And what if we could directly deliver this more to the plasma membranes, for example, like using cyclodextrin as a carrier and directly deliver it, as opposed to relying on these trafficking mechanisms. With regard to gene therapy, I think the challenge there is that, a lot of times we're using like adenoviruses, which depend on CME to get internalized. And that internalization is actually kind of the, like a key step in hindering the process. And so there may be other alternative routes, which is something that's done in other conditions where CME is known to be dysregulated. And you can utilize different pathways of getting into the cell. So that may be a different way that we could approach those challenges. So,
okay.
Yeah, thanks for asking.
Well, actually, thanks, Kevin, for that comment. All right. Any other questions? Okay, well, this was a fun day. And I have to say, I didn't realize we did it intention. I don't think we had this as our intention for a scholarship day, but I love the fact that we had all students present their abstracts today. And they show that the medical field in the future will do well because we have great people right now in training. And I want to thank the faculty, Kevin Francis, Michelle Bach, Benson Shu, Jennifer Shu. I'm sure I'm not covering everybody, for encouraging our students to follow research and scholarship. So with that in mind, thank you very much, and congratulations, Rebecca Maybe, for your award. And we look forward to the next scholarship day that we'll have next year, we hope, in person with posters and everybody explaining what they're up to. So thank you very much and enjoy the rest of the day. Yay.